Question number 1. Which of the following responses shows several hierarchical levels of biological organization in correct order? This is a very simple question, right? You know, molecules, they get together collectively, they form organelles, right? Organelles collectively form cells. Then group of cells form tissues, and one or more type of tissues form organs. Then organs collectively form organ systems and organism. So here for the first question, the answer should be third answer. Question number 2. Select the correct statement regarding carbohydrates. Now, if we take the definition of carbohydrate, right, you know, carbohydrate is the most abundant organic molecule, organic compound on earth, right? Major elemental composition is carbon, hydrogen, and oxygen, right? Yes, now look at the first statement. H/O ratio of all carbohydrate is 1:2. But in the definition, they have given hydrates of carbon contain the same proportion of H/O, right, which is equals to 2:1 as in water. Now, if we take water molecule, what is the chemical formula? H2O. So what is the ratio between hydrogen and oxygen atom? 2:1, right? So it is same in carbohydrate, in the hydrates of carbohydrates. But here it is given 1:2. So the first statement is incorrect. Third one, all carbohydrates are macromolecules. What are macromolecules? Right? There are two properties. Now, if we take a macromolecule, it should have a molar mass ranging from 10 to the power 4 to 10 to the power 10 daltons. And the second property is they are polymers made up of large number of repeating units, all monomers, right? Now, you know, carbohydrates in your syllabus, it is classified into 3 groups: monosaccharides, disaccharides, and polysaccharide. Right? There's another group. Under biochemistry, you can learn it later if you are interested in that area. That is oligosaccharides. Right? Anyway, it's not relevant to our syllabus. Monosaccharides, disaccharides, and polysaccharide. Oligosaccharide කියන්නේ, then disaccharide means 2 monosaccharides. Oligosaccharide means 3 to 10 monosaccharides, right? If there are 3 to 10 monosaccharides, they are considered as oligosaccharides, right? Then 10 වලට වඩා වැඩිපුර තියෙනවා නම්, if there are more than 10 monosaccharides, right, monomers, they are considered as polysaccharides, right? Here, all carbohydrates are macromolecules. But if we take monosaccharides and disaccharide, right, in disaccharide, there are only 2 monosaccharide units. So they are not polymers, right? Only polysaccharides are considered as polymers. So in carbohydrates, only polysaccharides, they are considered as macromolecules, not all carbohydrates. Fourth one, all carbohydrates possess glycosidic bonds. Now there are peptide bonds, ester bonds, hydrogen bonds, different kind of bonds. The type of bond that present in carbohydrate is glycosidic bond. What is a glycosidic bond? It is a bond formed by removal of a water molecule from 2 adjacent monosaccharides by a condensation reaction. Right here, water molecule, it is formed from OH group of 1 monosaccharide molecule and hydrogen from adjoining monosaccharide molecule. Okay. So here, all carbohydrates. Now, if we take polysaccharides, they are in polysaccharides, there are few hundred to few thousand monosaccharide subunits, right? They are bonded by glycosidic bonds. And if we take disaccharide, disaccharides also sugars formed by joining two monosaccharides by a glycosidic bond. Yeah, okay. Now, the glycosidic bonds are present in both disaccharides and polysaccharide, but there are no glycosidic bonds in monosaccharide. So, all carbohydrates, since they have given all, that statement is also incorrect. Then, fifth one, all carbohydrates are water soluble, right? Carbohydrates, we can classify them as sugars and polysaccharides. Here, sugars, monosaccharides and disaccharides collectively known as sugars, right? Sugars, they are sweet in taste and water soluble. But polysaccharides, they are not crystalline, right? They are not sweet in taste, and they are not, since they are not considered as sugars, they are water insoluble, right? So fifth statement is also incorrect. So the answer should be second answer, right? Genetic material of organisms contain carbohydrate. What is genetic material? DNA. In the backbone of DNA, you know, there are pentose sugar groups, right? Pentose sugar group. So we can take second answer as the correct answer. Then third question. Function of some organelles are given below. A. Conversion of fatty acids to sugars. That is done by glyoxysomes, right? Glyoxysomes. Then B. Production of transport vesicles. That is done by smooth endoplasmic reticulum and rough endoplasmic reticulum. Right, what is the difference between smooth endoplasmic and rough endoplasmic reticulum? Now, if we take rough endoplasmic reticulum, they consist of flattened sacs and there are ribosomes bound with surface. Okay, but in smooth endoplasmic reticulum, it is a network formed by tubular sacs. In rough endoplasmic reticulum, it is flattened sacs. In smooth, it is tubular sacs. And there are no ribosomes bound with membrane. But in smooth endoplasmic reticulum, there are membrane-bound enzymes. Right, what are the functions of rough endoplasmic reticulum? Right, in the resource book, there are 4 functions. First one, transport proteins synthesized by ribosomes, then synthesize glycoproteins. Third one, produce transport vesicles. Fourth one, facilitate the growth of all membrane by adding phospholipids, proteins, and carbohydrate. Right? Therefore, it is called as membrane factory. In smooth endoplasmic reticulum, they have given 5 functions. First one, synthesize lipids, including oils, steroids, and phospholipids. Then metabolism of carbohydrates. Then produce transport vesicles to transport within the cell. Then involves detoxification and stores calcium 2+ ion. Those are the functions of smooth and rough endoplasmic reticulum. C. Production of cell wall components such as cellulose and pectins. That is done by Golgi apparatus or Golgi complex, right? In the resource book, there are 3 functions given under Golgi complex. First one, collecting, packaging, and distribution of materials, right? Please use these words right there. I'm revising those things for the structured essay paper. Please write those given words, collecting, packaging, and distribution. Instead of packaging, don't write storage. They won't give marks. Okay. Then second one, manufacturing cellulose and non-cellulose cell wall components. Third one is produce lysosomes. Okay. D. Removal of residual materials from the cells by exocytosis. That is done by lysosome. There's another thing called lysozyme. It is an enzyme, right? Lysosome means an organelle, single membrane bounded vesicle, okay, which contributes to the digestive activity. These lysosomes, they contain hydrolytic enzymes which catalyze the breakdown of carbohydrates, proteins, lipids, and nucleic acids. What are the functions? What are the other functions? They digest food particles received by phagocytosis. So, macrophages වල එහෙම තියෙනවනේ. Then transport residual material out of the cell by exocytosis, digest worn out organelles, and autolysis causing cell death. Those are the functions. Now, in the question, what they're asking, functions of lysosome and endoplasmic reticulum are stated respectively, right? Lysosome, it's given in the answer D. And endoplasmic reticulum, as I told you, production of transport vesicles, it is common to both smooth and rough endoplasmic reticulum. So, it's given in B. So, D and B. Answer should be fifth answer. Then, question number 4. Right. A stage of cell division is shown in the following diagram. Now here you can see the centrioles, right? Microtubules, aster, okay? This kind of stage can be seen in the anaphase of mitosis and in the anaphase 2 of meiosis. So now here, look at the question. Which of the following statement is correct with respect to the above diagram, right? So it may be a diploid cell, yeah. I told you, like, if it is present in like, now this kind of stage can be seen in anaphase 2, right? Anaphase 2, it comes under meiosis. Meiosis takes place only in diploid cells, right? This also can be seen in the anaphase of mitosis. Mitosis takes place in both diploid or haploid cells, right? So it can be a diploid cell. It may be a cell of an angiosperm. Angiosperm means plants name. But here you can see centrioles. You know, centrioles, they present only in animal cells, but not in plant cells. So we can't take second answer. It cannot be a stage of meiosis. Incorrect, right? It cannot be a stage of meiosis. That is incorrect. It may not be a stage of mitotic division. Mitotic means mitosis. That is also incorrect. Daughter cells produced by this division are haploid, right? Yeah. Now, if we take this one, this kind of it produced by this division are haploid. In meiosis, it's a reduction division, right? Then if it start from 2n, then daughter cells will be n, right? And if it is a diploid one, no, if it is mitosis, n එකක් ආවොත් n වලින් නවතිනවා, right? 2n නම් 2n වලට යනවා. But the most suitable answer is the first answer. Question number 5. Effect of pH on the rate of reaction of two enzymes A and B is illustrated in the following graph. Right? This question is given from enzyme part. Now, this kind of figure, this same figure is given in your resource book, right? A, what is this enzyme? What is this enzyme? What is this enzyme? A should be pepsin, right? A should be pepsin. And B, it should be trypsin. Pepsin, it present in the stomach. Okay? Why? Any doubts? And B, trypsin, it is an intestinal enzyme. Now here, both the enzymes A and B can be functional within a single organ of an organism. No. A, it is present in the stomach and B, present in the intestine. Okay? Right, okay, I'll give I'll do an explanation about enzymes. Then stew, enzyme A and B could be trypsin and pepsin respectively. No, it should be other way around. A is pepsin, B is trypsin. 3. Rate of reaction of enzyme A at pH 1 and 3 could be more or less equal to the rate of the reaction of enzyme B at pH 7 or 9. We'll keep that answer. Let's go to fourth one. Optimum pH range of enzyme A is 0 to 2, while that of enzyme B is 6 to 8. Right? Optimum pH of A is 2 and optimum pH of B is 8. But there is no, they have not mentioned anything about something called optimum pH range. Range එකක් ගැන කියලා නෑ. Resource එකේ. කියලා තියෙන optimum pH එක. Pepsin වල optimum pH is 2 and in trypsin it is 8. So fourth statement is also incorrect. Then most of the enzymes of man are similar to enzyme B. No, right? These two are exceptions, right? These two are exceptions. So the answer should be the third answer. Enzyme, right? Now I'm going to explain about enzyme. We'll go through it quickly. Now enzymes, what is the definition? It is a macromolecule, right? It acts as a biological catalyst. Enzymes, they are produced in living cells. What are the characteristics? Most enzymes are globular proteins. Most, not all, most. Then they are considered as biological catalysts. So they lower the activation energy of the reaction. They catalyze and increase the rate of the reaction. Okay. But in chemistry, it's a different thing. Okay. Here, they lower the activation energy and increase the rate of the reaction. Third one, most enzymes are heat labile or heat sensitive. Most, not all, right? Fourth one, then their presence does not alter the nature or properties of the end products of any reaction. Enzymes are highly specific to the substrate. Then most enzyme catalyzed reactions are reversible. Okay, then they don't used up during the reaction. හරිද? catalyst එක used up වෙන්නේ නැහැ. එයාව ආපහු හම්බ වෙනවා ඊළඟ reaction එකේ අන්තිමටම. Then they have active sites. Active site is the place where that substrate go and bind to the enzyme and reaction takes place. Then some enzymes, they need non-proteinous components, right? They are known as cofactors to catalyze the reaction. Only some enzymes, not all. And the rate of the enzyme activity is affected by pH, temperature, substrate concentration, and also by enzyme concentration. Okay. Now, if we take the mechanism of enzyme action, first substrate go and binds to the active site of the enzyme and it forms an enzyme substrate complex. Okay. While this enzyme and the substrate form that complex, catalytic action of the enzyme converts the substrate to its product. Right? Then next thing, you can read those things later, right? Go through the resource book and you can study them. Then cofactors, as I told you, some enzymes, they need these cofactors. Cofactors means they are non-proteinous components essential for the catalytic action of some enzymes. Right? There are organic cofactors as well as inorganic cofactors. Organic cofactors, they are known as coenzymes. Examples are derivatives of vitamins, right? NAD, FAD, biotin, right? Vitamin B7, like that. Inorganic cofactors, zinc 2 plus, then copper 2 plus, Fe2 plus, ඔය ටික තමයි එන්නේ. ඊට පස්සේ factors affecting the enzyme activity. Temperature, right? Now, you know, when the temperature increases, what happens? It increases the kinetic energy or the molecular motion. Increase the molecular motion. And the speed of the kinetic energy increases. Mean the speed of the molecules, both the enzymes and substrate increases, right? So when their motion increases, what happens? The colliding probability of both enzyme and the active site and the substrate molecule increases. So there will be more collisions, right? And it generate greater chance for the reaction to occur. But it continue only up to a certain temperature that is called optimum temperature. හරිද optimum temperature එකට යනකම් ඔන්න ඔය විදිහට වෙනවා. මේ optimum temperature it varies from organism to organism. හරිද? Normally optimum temperature of bacteria that live in hot spring it is 70%, and in our body temperature it is 35 to 40 degrees Celsius. Right? What happens if we increase the temperature above the optimum range, optimum temperature? What will happen? You know, if we take the now enzymes, I told you they are globular proteins, right? There are different bonds, hydrogen bonds, ionic bonds, and weak chemical bonds. So if the temperature increases beyond the optimum temperature, those bonds, hydrogen bonds, ionic bonds, weak chemical bonds of the active site of the enzyme, they will be disrupted, right? Active enzyme, active site එක වෙන shape එකක් ගන්න. ඒකේ bonds කැඩිලා ගිහිල්ලා අරක මේ shape එක වෙනස් වෙනවා. I told you enzymes are highly specific to the substrate. එතකොට active site එකේ shape එක තියෙන්නේ substrate එකට ඇවිල්ලා ගිහින් අර ඒ substrate එකේ shape එකට තමයි තියෙන්නේ. හරිද? If the shape of the active site is altered, what happens? Now no longer these enzymes are specific to the substrate, right? Then active site එකේ shape එක වෙනස් වුණා. එයාට substrate එක ආවට ගිහිලා bind වෙන්න බෑ. ඇයි? දෙකේ shapes වෙනස්. හරිද? එතකොට මොකද වෙන්නේ? reaction එකේ rate එක අඩු වෙනවා. හරිද? The second thing is pH, right? There is a pH range in which the enzyme functions efficiently, right? එතකොට normally pH range, the definition of pH range is it is the narrow range of pH in which a particular enzyme catalyzed reaction takes place. pH range. pH range එක තියෙනවා optimum pH range කියලා එකක් අපි ඉගෙන ගන්නෙ නැහැ. හරිද? The alterations above or below this pH range, right? It decline the enzyme activity. එතනදිත් මොකද වෙන්නේ? pH එක වෙනස් වෙනකොට මේ chemical bonds ඒවා කැඩෙන්න පුළුවන්. ඒවා alter වෙන්න පුළුවන්. හරිද? ඊළඟට substrate concentration. Right? Increase in the substrate concentration increases the probability of collision between the enzyme and substrate molecules. Again, it generates greater chance for the collision, right? Collision between enzymes and substrate molecule. And next one, enzyme inhibitors. Enzyme inhibitors also affect the rate of enzyme activity. Okay. Enzymes inhibitors, there are two types as irreversible and reversible inhibitors. හරිද? ඔගේ අපි ඉගෙන ගන්න ඊළඟට competitive, non-competitive කියලා ඉගෙන ගන්නවා. Right? That is the outline about enzymes. In 2019, question number 6. Select the correct statement with regard to the C4 mechanism of photosynthesis. You know there are two types of mechanisms, C3 and C4 mechanism. Now this question is regarding the C4 mechanism of photosynthesis. First one, first carbohydrate derivative produced is a carbon 4 compound, right? In C4 mechanism, the first product is oxaloacetate acid, right? Oxaloacetate or AA. It is a 4-carbon compound, right? Now, if we take the C4 pathway, it is something like this. You know, there is something called Cran's anatomy. හරිද? එතකොට එතන තියෙනවා මොනවද? Mesophyll cells and bundle sheath cells. Right? In the mesophyll cells of the C4 plant, what happened? Carbon dioxide, it converts into bicarbonate by the carbonic anhydrase. Here carbon dioxide, then atmosphere එකේ තියෙන carbon dioxide mesophyll cell එක ඇතුළට එනවා. mesophyll cell එකේ cytosol එකේදී මොකද වෙන්නේ? That carbon dioxide binds with H2O and produce bicarbonate. Right? Here carbonic anhydrase, it acts as the enzyme here, and it converts into bicarbonate, and this bicarbonate is initially fixed by PEP, phosphoenol pyruvate carboxylase, right? Here PEP, it acts as the carbon dioxide acceptor, okay, and produce oxaloacetate. It is a 4-carbon atom, right? It is the basis for the name of this metabolic pathway. Sorry. Then this oxaloacetate, it rapidly converts into more stable 4-carbon acid, malate, or it's also called aspartate. Then it diffuses into the bundle sheath cells. Okay. Then in the chloroplast of the bundle sheath cell, right? This malate, it reduces into pyruvate, right? And then that Calvin cycle, it takes place. Calvin cycle එක මෙතනදී වෙනවා. හරිද? Yes. So first statement, it is correct. Then photorespiration does not occur due to the absence of Rubisco enzyme. No, that is incorrect. Third one, fixation of atmospheric carbon dioxide occurs within the cytoplasm of leaf mesophyll cells. Right? Carbon dioxide, please. Right here. Then fixation of atmospheric carbon dioxide. Carbon dioxide fix කරනවා කියන එතන Calvin cycle එකක් වෙන්නේ. So Calvin cycle, it takes place in the bundle sheath cell, not in the mesophyll cells. Okay. So the third statement is also incorrect. Calvin cycle occur in the leaf mesophyll cells under high carbon dioxide concentration. Right? Incorrect. Takes place in the bundle sheath cells. 3-phosphoglycerate is not an intermediate product, right? If Calvin cycle takes place in the bundle sheath cell, 3-PGA, 3-phosphoglycerate will be produced. So the fifth statement is all incorrect. So the answer should be first answer. Then, question number 7. The 3 eras of Phanerozoic eon in correct chronological sequence are. This question is given from the 3rd chapter, right? Now, if we take, there are 4 geological eons: Hadean, Archean, Proterozoic, and the Phanerozoic. And Phanerozoic eon, it can be divided, it covers 3 eras: Paleozoic, Mesozoic, and Cenozoic, right? So answer should be 4th answer. Now, Paleozoic, Mesozoic, Cenozoic should be 2nd answer. Okay. Next one. Question number 8. Carolus Linnaeus proposed system of binomial nomenclature of species which was accepted worldwide. Given below are the scientific names of some species. Right? If we revise that history of classification, Aristotle, he was the first to classify organisms scientifically. And he classified organisms as plants and animals, right? Then animals are further classified, right? ඉස්සෙල්ලාම animals ලා plants ලා විදියට බෙදුවා. ඊට පස්සේ සත්තුන්ව ආපහු classify කරා. What are the criteria? Right? Mode of locomotion, reproduction, and presence or absence of red blood cells. Second one, Theophrastus, right? He classified plants according to habitat as trees, shrubs, and herbs, and according to lifespan as annuals, biennials, and perennials. Then Carolus Linnaeus, right? He introduced something called binomial nomenclature. And he classified plants into a hierarchical order of taxa, right? Taxa classification levels. What are the 4 levels? Species, genus, order, and class, right? And his classification of flowering plants was based on the number of stamens and the styles of the flower, right? And he identified 2 kingdoms, plants and animals. Kingdoms දෙකකුත් අඳුන ගත්තා. හරිද? Then Ernest Haeckel, he introduced the 3rd kingdom, Protista, and the taxon, phylum. Yeah, taxon එකකුත් දුන්නා. Phylum කියන taxon එක. Then Robert H. Whittaker, හරිද? electron microscope එක discover කරගත්තන් පස්සේ තමයි scientists, they identify that there are 2 types of cellular organization, prokaryotic and eukaryotic. So after that, Robert H. Whittaker, he introduced 5 kingdom classification: Monera, Protista, fungi, Plantae, Animalia. Right? එයාගේ criteria ටික තමයි nature of the cellular organization, unicellular or multicellular, and the mode of nutrition. Right? අන්තිමටම Carl Woos, he introduced the 3-domain system. හරිද? Domain bacteria, archaea, and eukarya. දැන් අපි use කරන එක. Right. Now here in this question, Carolus Linnaeus proposed a system of binomial nomenclature of species which was accepted worldwide. Given below are the scientific names of some species. A. Dipterocarpus selenicus. Horror. හරිද? B. Homo sapien sapien. දැන් ඔයගොල්ලෝ දන්නේ Homo sapien කියන එක. ඔගේ දෙකක් තියෙනවා. Homo sapien කියන්නේ Neanderthalis. Homo sapien sapien කියන්නේ modern man. Right? Cocos nucifera. Right? Coconut. And then Panthera pardus cotia. Right, Sri Lankan leopard. Which of the above species name is so are in accordance with Linnaeus system binomial nomenclature? What are the rules here in the binomial nomenclature? 2 species of organism cannot have the same name, right? The scientific name should be composed of generic name and the specific epithet, right? අර දැන් exam papers structured එකේ මානේ genus කියලා අහන්නේ. එතකොට එතනදී generic name එක විතරයි ලියන්න ඕනේ. හරිද? Pogonato. Scientific name එක අහුවාම එතන specific epithet එකක් ඕනේ. Right? Then the name should be Latinized. It should be made up of the Latinized words written in Roman script, and it should be underlined if it is handwritten, and it should be italicized when printed. Right? The first letter of the generic name should be capitalized, and the other letters should be they are simple letters. Right? ඊට පස්සේ දැන් මේ බලන්න මෙතන. Now according to those rules, first one, Dipterocarpus selenicus, yeah, that is okay. It's printed, so it is italicized, right? First letter is capitalized, right? Yeah. B, Homo sapien sapien, that is also correct. C, Cocos nucifera, L. L කියන්නේ මේ Linnaeus. Linnaeus තමයි එක දීලා තියෙන්නේ. හරිද? L for Linnaeus. But here the letter L, it is italicized, right? It should not be italicized. L කියන එක කියන්නේ ඒක දුන්නු කෙනාගේ නම අන්තිමටම ලියනවා නම් ඒ කියන්නේ හරිද? Capital letter එකක් එහෙම නැත්නම් abbreviation, or it should be given as in full word at the end of the name, and it should not be italicized. Italicized කරන්නේ නැහැ. හරිද? එහෙනම් C වැරදි. D, Panthera pardus cotia. Cotia කියන එක italicized කරලා නැහැ. හරිද? එතන cotia කියන්නේ subspecies එකක් or a variety. Right? Here this cotia word should also be italicized. So D is also incorrect. Only A and B correct. Answer number 3. Question number 9. A trait that cannot be observed in the first land plants that evolved from the green algae. Right? Now, plants, it is believed that the members of this kingdom Plantae evolved from chlorophytes or green algae. Right? So, plants, they are terrestrial organisms. Now, these chlorophytes, so green algae, they lack key traits of these land plants. What are the key traits? Wall spores produced in sporangia, then multicellular gametangia, dependent embryo, and apical meristem. Right? Now, in this question, they're asking trait that cannot be observed. Right? First one, okay. Production of spores within walled sporangia, correct. Multicellular gametangia, correct. Presence of embryo that depends on the gametophyte, okay. Dependent embryo. Presence of apical meristem, correct. Formation of roots, that is incorrect. Now, if we take Pogonatum, it also belongs to kingdom Plantae. But are there roots? No, they have rhizoids instead of that, right? So, answer number 5. Question number 10. In which one of the following the invertebrate phylum and its characters are not correctly matched? Platyhelminthes, eye spots, and flame cells. Right? Yeah, eye spots they have. They have they have eye spots. Then flame cells for excretion. Flame cells කියන්නේ මොනවද? අර protonephridia. Right? Under kingdom Animalia, they have given as protonephridia. හරිද? ඊට පස්සේ excretory system එක ඔයගලන්ට දීලා තියෙන්නේ flame cell කියලා. Flame cell කියන්නේ protonephridia වලටම තමයි. Right? It's a specialized excretory cells connected to the network of the tubule which opens to the outside of the animal. Right? Then Cnidaria diploplastic and they have cnidocytes. Arthropoda exoskeleton and jointed appendages present. Mollusk, they have hemocoel and they have radula, minute toothed ribbon, right, that present in the mouth. Right? This hemocoel is present in both arthropods and mollusks. Now, nematodes, setae and cuticle, yes. Their body is covered by a tough cuticle, but setae, we can't take that one. So, the answer should be second answer. Question number 11. Which of the following statement is correct regarding the root of dicotyledonous plant? Right? Epidermis is multilayered? No, it is the outermost single cell layer. Then distinct pith is present? No. Pith is present in the stem, right? Not in the root. Collenchyma is present in the epidermis? No. Pericycle has the meristematic capability? Yes. Right? Dicotyledonous, in dicots, the pericycle has the meristematic capability, but in monocots, right, pericycle do not have meristematic capability. So the answer should be third answer. Question number 12. Which of the following external factors contributes to an increase in rate of photosynthesis and a decrease in rate of transpiration? Right? Now, if we take photosynthesis, it is affected by carbon dioxide concentration in the substomatal cavity of plant leaves, light intensity, temperature, okay? So, then water, pollutants, inhibitors, and if we take transpiration, it is also affected by light intensity, humidity, temperature, then carbon dioxide concentration, right? Then the availability of soil water, right? Now here they are asking the external factor which contribute to an increase in rate of photosynthesis and decrease in rate of transpiration. Light intensity, when the light intensity increases, what happens? The rate of transpiration increases. But here they're asking the decrease, right? Temperature, when the temperature increases, rate of transpiration increases. Then humidity, when the humidity decreases, rate of transpiration increases. Then available of water content in the soil, right? Soil water වැඩි වෙනකොට තමයි transpiration වැඩි වෙන්නේ. හරිද? Carbon dioxide concentration, substomatal cavity එකේ when the carbon dioxide concentration in the substomatal cavity increases, what happens? ඇතුලේ තියෙනවා නම් එහෙනම් ඒගොල්ලෝ ස්ටොමාට වහලා දානවා. වහලා දානවා කියන්නේ transpiration වෙන්නේ නැහැ. right? ඇතුලේ carbon dioxide තියෙන හින්දා එවෙන් photosynthesis වෙනවා. So the answer should be fourth answer. Right? We'll discuss the third. Now what's the time? It's 6.